Hi, it's Hosen here. Today I will show you how to enumerate bacteria by using this virtual lab. Before that, allow me to explain a little bit about bacterial enumeration. The meaning of enumeration in microbiology is the determination of the number of individual variable microbes in a sample, and this method is used to determine the number of bacteria by comparing the amount of bacteria growth under various conditions. But bacteria are so small and numerous, counting them directly can be very difficult. So the sample will be diluted and dyed to allow the observer to view the bacteria. For this video, I will only perform the variable plate count method. A variable plate count allows the observer to identify the number of actively growing or dividing cells in the sample. The colony becomes visible to the naked eye, and the number of colonies on the plate can be counted if the appropriate dilution is plated. This is the procedure for variable plate count. After the procedure part, I will guide you to count the number of colonies in our first sample, which is fountain water. First, click on the dilution tube rack of test tube to bring them to the table. Each of the dilution tube contains 9 ml of sterilized saline solution. Click on the water sample to bring the sample to the table. Now click on the eyedropper to withdraw 1 ml of sample 1, which is fountain water, to dispense this to the first dilution tube. Go to the next plate. Now click again the eyedropper to withdraw 1 ml of the first dilution tube to dispense into the second dilution tube, and subsequently withdraw 1 ml from the second dilution tube to dispense into the third dilution tube. Continue doing this from tube to tube until the dilution is complete through dilution tube 6. So we go to the next page. So the dilution for each of the 6 dilution tube can be summarized in this image. Dilution tube 1 has 1/10 dilution with a dilution factor of 10. So tube 2 have 100, tube 3 have 1000, tube 4 have 10,000, tube 5 have 100,000, and tube 6 have 1 million. So we go to the next page. Next, we have to inoculate agar plate with the last 3 broth culture, which is 4, 5, 6. So we click on the agar plate. By using the eyedropper to transfer 1 ml of dilution 4 to plate 1, 1 ml of dilution 5 to plate 2, and 1 ml of dilution 6 to plate 3. Next, click on the pencil to label agar plate 1 with dilution factor 10,000, plate 2 with dilution factor 100,000, and plate 3 with dilution factor 1 million. Go to the next page. Now click on the agar plate to place them into the incubator at 37 degrees Celsius for 48 hours. Then we go to the next page. Lastly, click on the incubator to bring all the inoculate agar plate to the table. Now click on the agar plate with number 1, which is the fountain water sample. So now we can count the number of colonies. So first, I will show you how to count the colony numbers in 10,000 dilution factor. So for this one, we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32, 33, 34, 35, 36. So we have 36 colonies in 10,000 dilution factor plate. So we continue with 100,000. So in this plate we have 1, 2, 3, 4, 5, 6, 7. We have 7 colonies grow in this plate, and continue with the last plate. In this plate we only can see 1 colonies variable in this plate. So that's all for today's video. I will put this virtual lab link in the description section. You can do it if you like. Thanks for watching. Bye bye.